It is my honor to introduce our eminent speaker, Dr. Rajiv K. Tyagi. Sir has earned PhD degree at Biomedical Parasitology Unit in the prestigious Institut Pasteur, Paris, France in 2011. On a very challenging and interesting topic of malaria, immunology, and parasitology, he developed a long-lasting, stable, and straightforward laboratory animal study pathology and immunology of infectious diseases and beyond. Sir has worked as a postdoc fellow in laboratory of Dr. John Adams, University of South Florida, USA, and has received training to explore the potential of the developed humanized mouse to understand the innate immunity response of the attenuated plasma. Hello, can you hear me?
Yes, yes, Dr. Neil, please go ahead.
Okay. Sir has received postdoc fellow in the laboratory of Dr. John Adams, University of South Florida. Understand the response of attenuated Plasmodium falciparum parasite, among other works. Sir, at the Department of Medicine Division of Gastroenterology and Nutrition at Vanderbilt University of Medical Center in USA deployed his efforts to understand the role of interleukin 23R in modulation of functioning of regulatory T cells. Sir has published more than 50 research articles, 9 book chapters, and edited 3 books, and has a very impressive citation score of more than 1300, H index of 21, and I-10 index of 31. Currently, Sir is a leading a group of CSIR Institute of Microbiology Technology, Chandigarh, India. His lab is focused on mainly Plasmodium falciparum parasite and translational biomedical research, developing therapeutic vaccines. I welcome you, sir. Session is of 45 minutes of talk and 15 minutes of Q&A session. Now the stage is yours.
Thank you, Dr. Gupta. Thank you very much. I appreciate your efforts and very encouraging and stimulating introduction of mine. Thank you very much. Let me share the screen first. Okay. So I think I'm audible to all of us, right?
Yes, sir. Yes, sir.
Okay, that's great. That is great, actually. So, Dr. Ujjwal, as you mentioned earlier, also that we have the audiences of all the students, even some postdocs, as well as some, medical doctors and some faculty as well, right? Am I right? Is that?
Definitely, we have a wide range of
Audiences. Yeah. So today, this is my earnest request to all of my audiences and students, especially the young students, master students, and faculty, and all them PhD students as well. That this is let's say it's a sort of informal, I would say, the talk, and I am gonna walk you through that how to write an impressive abstract, okay, an introduction. So, and there is no hesitation at all from it should not be any hesitation from your side. You can call me anytime, you can ask any question, right? So I won't waste much of your time. As you can see that the very, I would say, very good webinar organized by this IAMBSS, which titled as Structure of a Scientific Articles and then Abstract Introduction Keyword. This is my topic today. That I'm going to walk you through to write abstract introduction and just to help you think that how to choose important keywords, right? So as Dr. Gupta has already introduced myself, so I'm currently, before starting anything, I would like to tell you, I'm like you guys. I'm amongst one of you people, and I am probably one of the youngest students of sciences. I'm still in the learning phase, and it's gonna take a while that I will develop as a scientist. Okay. So today, let me first start with research paper abstract. How to formulate a research paper abstract. Before starting anything, I would like to tell you that my talk will be consisting of two different aspects. Like first, in the first half of my talk, I'm going to talk how to write an abstract, what should be the way of writing, how to make it impressive. And in second half, I'm gonna take some examples of my own research, just to tell you that how to make it more impressive, okay? So you have to be little bit patient about that. So first of all, abstract. You know, abstract, in the very first thing, actually, I would like to tell you, abstract is a reflection of your introduction and especially the discussion, right? This is the reflection of introduction, especially the last part of the introduction, and then discussion. This is the reflection. So abstract are also divided into 2 or 3 different aspects. But since we are from, you know, experimental sciences, especially the life sciences, so I'm going to talk about those abstracts which are written for life sciences. So I'm using the word life sciences, and life sciences covers everything, right? Like all the disciplines, by especially for the young students, for my master students, for their information, that abstract life science covers everything, like biotechnology, microbiology, whatsoever you name, and it's a long list. So abstract, as I mentioned, it is the essence. I hope you all understand what is the meaning of essence. Essence, it's a gist of what the work is being carried out and carved out, right? So abstract further divided into 2 different categories. One is informative abstract, and another one is a structured abstract. Let me tell you what is informative abstract. Informative abstract means, and by and large, informative abstracts are actually not structured. Just to be very sure, means structured means that for the, as I mentioned, that abstract are actually the reflection of introduction, discussion, and to an extent materials and methods, that what the methodology you have adopted. But in by and large, abstracts are informative in nature, means there is no further subdivision. For example, in a structured abstract, as you can see in the, on screen, on structured abstracts are essentially, they are like divided in 3 or 4 different categories, like background information, then methods, results, and conclusion. But this structured abstracts are asked by many journals or many scientific forums just to formulate. Normally, it is informative. Informative means the abstracts are written in a flow, and the abstract is divided in 3 different categories. Number one, the first half of the abstract actually, it gives the reflection of the background. Now, what is background? You need to understand what is background. Well, so background, as I mentioned in the first half, in the beginning, the background, methods, results, and conclusion. This is the composition of all the abstracts, whether it is structured abstract or it is informative abstract, right? So now let us come, background is the most important thing, that whatever has already been done about the work that you are going to present in, so that is the background, or you can say the introduction. Background means the introduction, that the work you have carved out, that what else is already known about that particular work that you have carved out or you have conducted. So that's in the first point, you can clearly see that what is already known about the subject, that your paper relates to, right? That is what is already known. So that is the background information, and for that you have to do an extensive search, and there are many search engines available online. And nowadays all the students are pretty smart, and I accept this with very very humbleness and with honesty, that all the students nowadays are intelligent enough to search and to know more about the particular subject. Second point is that in the background information, in addition to what is already known, so in addition to that, you have to instill the information that is not known, or that you want to to give the information about. So that is another part of the background. And as I mentioned, abstract is divided in 3 to 4 parts. And the first part is background. Background itself contains 2 points. Number one, that is what the information is already known. And second point, that is not known, but you intended or you want to conduct the experiment on, or you have conducted the experiment on. So this is the second part of background. But please understand, don't confuse background. It should not be pretty long, and it should be maximum 3 to 4 points. 3 to 4. In fact, 3 to 4 lines are too much. I would say 2 to 3 lines, and that's it. Background is done. Now, I won't read whatever has been written on my slides, but what I'm saying, lengthy backgrounds, if you are writing a lot of information about background, or I would say the introduction in abstract, it's not good. It doesn't deemed as good information, that you should not write a very lengthy background. What I mean to say here is that lengthy background should be only a couple of lines long, and that's it, you are done. So another part, what I want to say, the second part of the abstract, as I mentioned, the first part, the first couple of lines should be about the background. The second portion is important as well, that what methodology, what the scientific question you have been addressing, and in order to address that scientific question, what the methodology, what are the techniques you have adopted or you have used, right? To be in a very simple language. So method section, especially method section in abstract, it can contain, it may be I would say longer than the first part, means longer than introduction or background information, but shorter than the result section, which will come later on. In the second half, we are going to talk about the result section. So first part, background part of abstract, the second part will be the methods part, and that should be written, composed in a very, I would say in a very legible manner. So that legible meaning that all the eligibles can understand, but even legibles who are not, who do not belong to that particular field can also understand what exactly the abstract is speaking about. So now, second, this will be the methods part, second part. But again, I'm using the word reflection. The abstract should have the reflection of material, methods and materials, but not exactly that you are going to write that so and so microliters you have added in that. No, you cannot write that in abstract. The abstract is only the reflection of what methods you have adopted. So you have to be intelligent and smart enough to write those couple of lines, or maybe more than couple of lines about methods in your abstract section. Third part, the most important part of an abstract is the result section, because whatever the scientific question you have addressed in your findings, in your discovery, where you need to compose in such a manner that it should comprise of, it should be instilled with all the information what you have done in a very, very scientific language, right? Because here we are talking about scientific language, not the English writing, etc. No, English is not a question here. Of course, you should have your vocabulary should be okay for that, just to write a result section or just to write a paper. So in that result section also, as you can see on the slide actually, that readers should be able to understand what exactly what you have done, and what findings, whether those findings are in coherence with the others' finding, means whether those findings, whatever has already been done, is in coherence with those findings, or some absolute novel finding, because findings are also categorized further. Finding number one, demonstrative finding. Number two, explanatory finding. So demonstrative finding is that those findings which demonstrate something but are absolutely not novel. Means if you because there are many students, young students among you people, that that there are big long list of journals. So there are certain huge journals, high ranking journals, which publish only novel data, novel finding. So it means they are standalone finding, means they have some novelty that those sort of findings has never ever been published. So these are absolute novel findings. But there are some demonstrative findings, means these findings demonstrate something which has something which has already been published. Not published, I would say, which has already been known. at least you can say. So this is the absolute demonstrative finding. There are certain explanatory finding and there are certain absolute novel findings. So demonstrative, explanatory, and novel finding. There are three different categories of any result section. So this is how. And the last part, last probably but not the least part, the conclusion part, means in couple of lines, only 1 or 2 lines, you need to conclude what your findings are all about. What is the take-home message? Means what is the take-home message? Means what a reader can take a message home, right? It means you need to be very, very expressive in this conclusion section, that what your findings are on, what your findings are related to, and based on your findings, excuse me, based on your findings, what your perspective will be, future perspective will be, means from there how the research can be taken forward. So I would conclude this section. Number one, I would tell you the abstract is a part of 3 different sections. Number one, background information. Background means known information as well as unknown information. The information which is already available, and unknown information, the second part of the background part, that is that the second part is that the information is not known in the public domain. Second part of abstract is the methods part, and methods is again is very important and probably the second longest part of the abstract. That is that you need to fill in, or you need to fit in all the information and methodology what you have adopted to conduct your experiment, to finish off your experiment, just to get the desirable results and amenable results. Amenable means, I don't know whether you know, many people you may be knowing about this. Amenable means those data which are statistically significant. This is amenable data. And the third part of the abstract is the result section. The results, as I mentioned, it's a very important section and the longest part of the abstract. Correct? And result, as I mentioned, result also further categorized in 3 different categories. Category number 1, the demonstrative results or data. Then second is explanatory data. So these are 2 or 3 different sections of the result part also, which comprises of your abstract. And last part, the concluding remarks that you want to give, that what your study is all about, and from there what can further be done. So this is all abstract. If someone, I don't know whether if someone is having questions or at the moment, or maybe I'll answer all the questions in the later half. So second, well, this is another, I would say, I have written something from somewhere that about the take-home messages. As I mentioned, that I told you very clearly, the take-home message is that what your study is all about. That is the primary message. Means your study, what your study, your findings are all about. I mean, what they have actually highlighted upon. And second, the additional findings, means in addition to the important finding, if someone additionally you have your findings are highlighted upon or highlighting on. And third, as I mentioned, the conclusion part, that what will be the future perspective from there, means your study can further be taken upon, and how the further can be taken off from there. What study, how your study is in coherence with other study which is already in the public domain, or maybe is in progress. So these are the part of the abstract. The first part, background part. Second part is the most longest part. That's the second longest part of abstract is the methods part, and the third part is results, and probably the last very, very crispy or one line message is the conclusion part, which is having 2 different take-home message. Primary take-home message, I would say, that which is very important from your finding or out of your finding, and then additional finding of your work. So this is all about the abstract section. Up now, as I mentioned, that I talked about abstract section, but now since Dr. Gupta has already informed me that I'm a parasite immunologist and I conduct immunology, so I'm gonna So I'm gonna talk about, I will take an example of my abstract or my finding, and then I will try to explain to you people. So I'm not very sure about it, whether I'll be able to make you understand, but I will try to use as much simple language as I can, right? So the title of one of my published papers, which is already out in public domain, that is in scientific research, Nature Scientific Research paper, which is already out there. So the title of our published paper is Human IDO Competent Long-Lived Immunoregulated Dendritic Cells, and its role, it's under the persistent intracellular pathogen and its role in humanized mouse model. It's tracking in humanized mouse model. So I'm not very sure again that how many of you people know what are humanized mouse models and stuff. But anyways, I'll just try to touch upon, and then we'll see. So this is the example of a I won't say it's a simple abstract, but it's a complex biological problem which I try to make it simple in a simple scientific terms. So there are, our immune system. Our immune system is having two different branches of our immune system, that is innate immune system and acquired immune system. Innate meanings, as its word implies, that innate means a sense birth, the immunity which is present in our body since we come out to existence in this material world. All right. So this is the innate immunity, and then acquired immunity. Acquired immunity means that during your lifespan, you are going to gain the access, you are going to gain the resistance to all the diseases. That is immunity means. All right. So immunity, I will very simple, in a simple language, any pathogen comes and you are afflicted with by any disease, then our innate immune system absolutely provoked, and it does it work. It clears out the pathogen and throw it away, right? This is the standard stuff. But when the pathogen, any bacteria, any parasite, or any other microbes is able to breach that barrier, or I would say that resistance, which is conferred by the innate immune system, then the acquired immune system comes into play, because innate immune system is not able to overcome those parasites, bacteria, or those microbes. All right. So in that case, acquired immune system means those B and T cell will come into play. And then if any microbes attacks your body, and then there are certain cells which are, I would say, the connecting link of acquired and innate immune system, and these are called as dendritic cells, or you can say in simple language, people say dendritic cells. So dendritic cells are, I don't know how many of you know, probably you know the monocytes and macrophages. Monocytes are the primary immune defense effectors, I would say immune effector cells. So monocytes, when they circulate in the blood, these are called monocytes. But as and when they enter any tissue, then those tissue specific monocytes are termed as macrophages, right? And macrophages are 10 times to the size of monocytes. So those macrophages, or would say that tissue-specific monocytes are macrophages, and macrophages when further differentiate into presentation cells, these are called as dendritic cells. Up now, in immunology language, how monocytes and macrophages or dendritic cells are differentiated based on the morphology, based on the structure.
Sorry to interrupt, sir. Actually, we're deviating from the topic, sir.
Okay. Okay. I'll say, actually, I got your point, but what I'm saying, I just wanted to, if I'll speak about dendritic cell CD14, then it will not help at all. Okay. But that's what. Give me just 5 minutes. I'll finish it off very quickly. So I was just setting the background actually. So because monocytes and macrophages and dendritic cells, this is the essence of this study. So now dendritic cells, monocytes and macrophages, 3 different stages. Everyone knows monocytes and macrophages. So dendritic cells are those cells which present the pathogen to your immune system. Just to clear that out, they're very simple. I'll use a layman language, right? So dendritic cells present and then process those antigens, because any foreign particle coming into your body and infecting yourself, these are antigens. So antigen are presented by those dendritic cells. So that this study revolves around dendritic cells. But I'm telling you, there are many categories, but I'm just going to speak about two different categories. One are monocyte derived dendritic cells, another one is pathogen derived dendritic cells. So the story revolves around here. So that is what you can say here. So monocyte derived dendritic cells, and another one, as I mentioned, pathogen, means monocyte derived dendritic cells need some stimulation, that is GM-CSF and IL-4, and then those monocytes differentiated into dendritic cells like phenotype here, IAMBSS you can see here. But another category is when there is a bacteria, this is called Porphyromonas gingivalis, you take them and they're isogenic, the different modified mutants. I will use very simple language for just to make students understand that they're modified I would say modified strain. When you incubate those modified bacterial strain with monocytes, they will differentiate without using any IL-4 or GM-CSF, any growth factor or interleukin. They will differentiate into pathogen differentiated dendritic cells, PDDC versus MODCs. So the whole story was about this, that how come the PDDCs, pathogen differentiated dendritic cells, they will allow this bacterium to reside for longer duration in your mouth cavity, despite the fact that the innate immune system is intact. So the scientific question was, this was the scientific question when I addressed that how come it is possible, despite the fact that your immune system is pretty intact. So now, just in order to address that, I just came out of my findings that there are only two different conditions, which one is apoptotic condition. Apoptosis, I'm sure you may be knowing, most of you may be knowing, apoptosis is programmed cell death, or you can say the death which is programmed. And another one condition is survival condition. So I will make it very short now. So, I'm not going to go into deeper into the immunology, otherwise you will be lost. So what I'm saying, those pathogen differentiated dendritic cells induce survival condition, and MODCs, monocyte derived dendritic cells, they were driven by apoptosis, means pathogen was smart enough to take a control on the on dendritic cells. Those are MODCs, monocyte derived dendritic cells, and that's how the pathogen, the bacterium, can reside for a longer duration in our mouth cavity. So this was just I just told you very simple, crisp of our study, that this was our study, and then this is how I could make a presentation out of that work. That is why I told you the background. Why? So this is why the first part, if you see, the first part was ex vivo generation of MODCs, monocyte derived dendritic cells, and pathogen differentiated dendritic cells. And if you can see, conventional mode means, as I mentioned, conventionally the whole world is preparing dendritic cells like this. So this is how the abstract can be graphical abstract. This is the way of graphical abstract, because any journal, if you are going to shoot a paper to publish in, it's going to ask you two different things. One is the descriptive abstract, and another one is the graphical abstract. And this is the descriptive abstract. That means that you need to write like 150 and 250 words. Because there are different journals, and their recommendations are different also. So it ranges, the word limits ranges from 150 and it goes up to 300 words. The higher ranking journals, they ask always 150 words. But there are, again, I'm telling you, it's a range starting from 150 until 300 words. So this was the first one. And then second one, I will just very simple take an example, again, very simple. That will take those lipid-based nanoparticles, and then how lipid-based nanoparticles entrap the drug is entrapped into them, and then treated those in an experimental model. Means we created an experimental model of rheumatoid arthritis, which is in Hindi called.
Sorry, sir. Sorry to interrupt once again, sir. There are a lot of people from physics background, and they may not be knowing about these monocytes.
Right. So, I understand. Okay. So let me then go to the next section. This is what I can do, right?
So you can explain about that visual abstracts and what is required for that.
Right. This is okay. So that's what if you see the first slide, in this slide, I already take an example that abstract are out through 2 to 3 different categories, right? The demonstrative abstracts and one minute. So abstract, as I mentioned, is three different categories. It divided in 3 different sections. Section 1 is background information. 1 minute. Background information, then it in methods, results, in conclusion part. So this is divided into 4 parts of the same thing, which is in descriptive nature, same applies to the graphical abstract also. Right, Dr. Gupta? This is what I wanted to convey my message about. All right.
Yes, sir.
So, but yeah, because this is, I can understand that your audiences may not be well-established people, but there may be certain other people also, right? So, but anyway, I'll just make it short. So, in brief, but I'll take 3 examples in brief quickly. I won't take much time. Maybe next 3 or 4 minutes, I'll finish it off. All right, this section at least. So in another part, there is another disease, because model disease you need to take about some model disease. If you are working on any disease, for example, rheumatoid arthritis, which is in Hindi is termed as Gatia Bai, and everyone is affected. Most of the aged people are affected with this disease. So we just wanted to address this disease because this is an autoimmune disease and no one can treat this disease. This disease, the pain can only be alleviated using, or the lifestyle can, or maybe the life standard can be increased, or lifestyle can be increased, can be enhanced by using our formulation. So we prepared formulation, and then we try to, to address this disease. I'm reluctantly using the disease word address, but I can use the word that alleviation of disease condition. So this was another example, and well, I won't tell you much things, but I don't know how many of you know that humanized animals which are used, those animals which appears like humans in terms of their immune system, because the mouse's immune system has been replaced by the human immune system in order to create a human-like conditions so that the human diseases can be harbored in those animals and can be studied. So in that sense also, that humanized animals can be used, and this is what I wanted, that how, and this was the abstract for a review paper, because as I mentioned, the papers are also categorized in 2 or 3 different categories. So paper, for example, the original research article based on your findings, and review paper based on your research, whatever you have done for last 10 or 15 years or so, that you want to review your papers. So this is the abstract for a review paper, and why I wanted to tell you about this actually. So for that also, that there in review paper, you are not going to write your findings, but you are going to conclude, or you in a very crispy language, out of 10 to 15 years what you have done, you wanted to just to review your findings in coherence with others' findings. So this is the abstract for a review paper. Well, now the another point is abstract. The last part of the abstract is the key, not abstract. I would say this is the last part of any manuscript. That keywords. This is very important, because based on the keywords also, the people will come to know that what exactly your manuscript is all about, because the keywords should be the central words of your manuscript. All right, means those words which are used multiple times in your manuscript, all throughout your manuscript, those words which are multiple times which have been used, that you need to pick those words up, and then at least 6 to 10. Whereas normally it is 5 to 7 words, but I would say 6 to 10 words would be good enough. All right. So in that case, and more, but if any reader is gonna read your abstract and your keywords, so he or she should be having every information that okay, this manuscript is about this stuff. So that keywords, abstract, and the title also, I would say that should be the that should give him or her an idea that okay, this is the finding, and this is so that on its simple glimpse, on a simple reading of abstract, he or she will understand. Here I would like to tell you one more thing, that if someone is gonna break into a very good journal, so the keywords, abstract, title, which should be very much appealing title, is very important if you want to open the window. And because once you submit your paper, that paper will first go into the editorial panel, and editorial panel will go through the abstract and your paper, and just to understand what exactly your work is all about. So it means this is very, very important. So keywords, abstract, and title, which should be very much appealing title. Like I'm telling you, the title should be picked up from somewhere from your discussion. That whatever you have discussed, the title should be picked up from there. I mean, not exactly the line you pick up from there, but you need to make it very much appealing. So abstract and title is very, very important, not only for your readers, but readers. And then if you want to publish a paper in a very respectable journal, then you need to be able to open the door for journal, good high ranking journals. Next thing is that, just a minute. Now let's talk about introduction. So how to write an effective introduction? Well, I'm just thinking how to start and where to start from. So let me tell you, introduction also, I'm not reading whatever I have written in the slides, but introduction, it should be very compelling. Means once any reader reads 3 or 4 lines, any readers reads 3 or 4 lines, so he or she should understand that okay, this should be the gist of the paper. So of course it should be in a simple language, especially the present tense it should be, and any abbreviations for any word which should be abbreviated, but for the first time it should be in its extended form, and thereafter you can use those abbreviations. And as I mentioned in the beginning, in the abstract section, also the reference section, you should be able to search out the literature, the relative literature, and of course you should be very clear. You should not be, and there should not be any ambiguity, and sentences should not be very long. They should be readable, and even legible should understand all these things. And you should address the issue. So introduction can be divided in 3 different parts. Number 1, the first part will be the background, as I mentioned in the abstract. Second part will be again the methodology reflection will be there. There should be the methodology reflection in the introduction as well. Sentences should be very short. And the last part of the introduction is very, very important. At least 4 to 5 lines, or I say 5 to 6 lines, that those are very important, that you should be able to express yourself in scientific terms, that what you have done and what is your finding and what may be your future prospect of your finding. Of course, I mean, as I mentioned, that for those 5 to 6 lines should be aimed at defining your goal, what you have done in your work. So this should be the composition of any introduction, effective introduction. That should be in a very simple language. And if you ask me personally, because I have published many articles, but I would say you should write an introduction as if you are talking to someone, as if you are talking to someone, and as if you are telling the story to someone. So this is the way you should write. And any complex biological problem should be expressed, should be expressed and written in a scientific language, but in a very simpler manner, so that all should understand what exactly you have done. So although I think Dr. Gupta, am I running out of time? Hello?
Yes, sir. Time is very, it is a constraint, sir. So, but
Give
please
me,
continue. Please continue, sir.
go give me only 2 or 3 minutes. So about the introduction, I wanted just to tell you people how to bifurcate this introduction in 3 different sections. Again, I'll tell you the same, one of my, works that where I developed nanoscale drug carriers and entrap the drugs, those aceclofenac and methotrexate drugs, and then deliver the drug and then try to address this problem. So in the first section, I just take an example from my work, which is under consideration in a very huge journal. All right, the first part will be the preparation of the background. If any one of you want these slides, I'll share all of you. Don't worry about that. If any one of you want. So as you can see in the first couple of lines, you see the I have written some few lines about rheumatoid arthritis. It's heterogeneous nature, and what the information is already available about this arthritis. As you can see that this disease secretes these interleukins, cytokines, etc., which is already known in public domain. So this first part, if you can see here, I have written a couple of lines about the disease, its etiology, its nature, its autoimmune in nature, and then treatment strategies which are already in public domain, because there are strategies like monotherapy and co-therapy, monotherapy and combination therapy. And in this is the background part, and for that you have used very latest references. For example, if you are living in 2021, so I think there are certain references which are very important because they are the mainstay of that particular finding. Then you may use those reference, whether it is 1978 reference or 2015 reference. But otherwise, the references should be taken from last 5 to 7 years only. That's it. In my paper, if you see that, I think I haven't given the complete list of references, but yeah, these are all 4 to 5 years later old references. That's it. So second part is, if you see here, I have clearly mentioned that I would say the reflection that what the methodology you have already used about the work you have carried out. So as you can see that there's a different pathways, signaling pathway, etc. I have mentioned. Second part is this, that as in introduction, as I mentioned, or in abstract, because abstract and in introduction, there is no difference between abstract and introduction, only the difference of length. And the third part will be the indication what you have proposes, what your work is going to propose, means what were your aim while you started your work. You finished your findings, and now this is all the goal of the aim of the study, and you have found out so and so stuff. Actually, like in our case, our finding revolved around co-therapy. Co means combination therapy of aceclofenac and methotrexate, which actually regulated all the immunological pathways, signaling pathways, and that's how it actually put a tab on the inflammation, because this disease is of inflammatory disease. So this is the indication of your, I would say, results and aims, I would simply say. Because last part, like 4 to 5 lines or 6 lines should be very, very important. And they should be written very carefully, because those lines comprises of all the gist of your findings, what you have done. And that's it from my side. I'm sorry if I have taken more time, Dr. Gupta. Sorry for that. And if there is any questions or concerns, I'm there. I will be available through email also. And if you want, I can share all the slides with all your audiences. Thank you very much.